previous class we completed up to enzymes. Now today we discuss about photosynthesis, the most important chapter of the second year plant physiology syllabus. Synthesis of food in the presence of sunlight by combining two complete inorganic components like carbon dioxide and water as chloroplast as site. Preparation of glucose and liberation of oxygen is called as photosynthesis. Photosynthesis is a physico-chemical reaction where physical changes occurs in the presence of light, hence it is called light reaction. Chemical changes occurs in the absence of light, which is called as dark reaction. It is an oxidation reduction reaction where water molecule get oxidized. Water molecule get oxidized to liberate 2 electrons, 2 protons and half O2, whereas carbon dioxide reduced to the level of carbohydrates. Now, the what you call carbon dioxide has come from atmosphere, water comes from the soil, sunlight is the ultimate source of energy. The first step in photosynthesis is excitation of chlorophyll. Chlorophyll remains in the excited state just for 10 to the power of minus 9 second or 1 nanosecond or 1 billionth of a second. Thus time is over for the electron transport. Then sunlight, chloroplast, appraised region, non-appraised region with pigment system 1, pigment system 2 and other aspects. Then light reaction with photolysis of water or Hill reaction, emergence enhancement effect, electron transport, proton translocation and photophosphorylation, formation of ATP in the presence of sunlight. One it is called non-cyclic, the other called cyclic for the formation of only ATP molecules. After that C3 cycle, C4 cycle. Factors controlling the rate of photosynthesis: light, carbon dioxide, temperature, then water, oxygen. Effect of oxygen on the rate of photosynthesis is called Warburg's effect. Then etiolation, internal factors etiolation. A plant grown in darkness definitely show pale color, poor growth of leaves and also very elongated stem. This is called etiolation. Then accumulation of end products. Then photorespiration or C2 cycle. Photorespiration is liberation of carbon dioxide even by the plants in the presence of sunlight. And finally, Blackman's law of limiting factors. So this is the chapter and this is the syllabus. Now we discuss some important bits in this chapter. This is an assertion. Oxygen is the byproduct of photosynthesis. Oxygen is a byproduct of photosynthesis. Whenever photosynthesis takes place, 6CO2 plus 12H2O in the presence of sunlight and chloroplast as site, C6H12O6 plus 6H2O plus 6O2. So, oxygen is the byproduct of photosynthesis, correct, because carbon dioxide, the ratio of carbon dioxide and water mixed is 1:2, when 6 carbon dioxide, 12 H2O. In the presence of sunlight and chloroplast as site, glucose is formed, metabolic water is formed and oxygen is liberated. So, oxygen is the byproduct of photosynthesis. Photolysis of water is the source for oxygen evolution that occurs in the beginning of light phase of photosynthesis. First step in light phase itself is photolysis of water or Hill reaction. Hill proposed that whenever a chloroplast is isolated and sunlight is exposed on it, the conditions are complete absence of carbon dioxide but in the presence of a hydrogen acceptor. The hydrogen acceptor used by Robert Hill is potassium ferric oxalate. In these conditions, the chloroplast which is isolated and exposed to sunlight liberates oxygen. In the complete absence of carbon dioxide, it is liberating oxygen, means definitely it is coming from water. So, oxygen is the byproduct of photosynthesis. Photolysis of water is the source of oxygen evolution that occurs in the beginning of light phase of photosynthesis. So, both A and R are correct. R is the correct explanation of A, first option. This is the match the following: chlorophyll A, chlorophyll B, pheophytin, xanthophyll. Chlorophyll A chemical formula is always C55H72O5N4Mg. So, second option. Chlorophyll A is always C55H72O5N4Mg. Chlorophyll B C55H70O6N4Mg. C55H70O6N4Mg. This is option 4. What is the difference between chlorophyll A and chlorophyll B means? In chlorophyll A and chlorophyll B, it lies in the third carbon of second pyrrole ring. Third carbon of second pyrrole ring shows the difference. Suppose third carbon of second pyrrole ring shows methyl group, it is chlorophyll A. Suppose third carbon of second pyrrole ring shows aldehyde group, it is chlorophyll B. So, chlorophyll A, chlorophyll D, difference lies in the third carbon of second pyrrole ring. If methyl group is present attached to the third carbon of second pyrrole ring, it is chlorophyll A. If aldehyde group is present in the place of methyl group, it is chlorophyll B. So, C55H70O6N4 Mg. Phaeophytin. Phaeophytin is a photosynthetic pigment which takes excited electrons from PS2 in non-cyclic photophosphorylation. In the textbook, it is given as it is almost resembling chlorophyll, but central magnesium is replaced by 2 hydrogen atoms. Central magnesium is replaced by 2 hydrogen atoms. So, its formula becomes C55H74O5N4. Mg is not there. 2 hydrogen atoms are extra there. So, definitely C55H74O5N4. Answer will be 1. Then xanthophyll. Xanthophyll is C40H56O2. Oxygenated hydrocarbon is xanthophyll. Carotene is C40H56. So, chlorophyll A C55H72O5N4 Mg. Chlorophyll B C55H70O6N4 Mg. Phaeophytin C55H74O5N4 H74O5N4. Xanthophyll is C40H56O2. Here the third option C40H56 is the what you call carotene. Now, wrong match in the following. Wrong match in the following. Chlorophysi contain chlorophyll A and chlorophyll B, because chlorophyll A is present in almost all photosynthetic plants except in photosynthetic bacteria. So, chlorophyll A and chlorophyll B. Phaeophysis shows chlorophyll A and chlorophyll C. Chlorophyll A will be the reaction center for any photosynthesis, chlorophyll A and chlorophyll C. Rhodophysi contain chlorophyll A and chlorophyll D. Rhodophysi contain chlorophyll A and chlorophyll D is correct. Cyanophysi contain bacterial chlorophyll A and bacterial chlorophyll B is wrong. Cyanophysi contain chlorophyll A and chlorophyll B only. I don't say chlorophyll B, it is chlorophyll C. So, fourth one is the wrong match of the following. Next, all stromathylakoid membranes are considered as non-appressed regions. All stromathylakoid membranes are exposed to stroma matrix. Any portion of stromathylakoid, ends of grana, margins of grana, suppose they are exposed to stroma matrix, they are called non-appressed regions. The portions of grana which are present as stack of plate-like structure not exposed to stroma matrix are called as appressed regions. So, all stromathylakoid membranes are considered as non-appressed regions is correct. Stromathylakoids are not piled one above the other. Stromathylakoids are not piled one above the other, or at some points only they are piled, and that portion is called grana, granathylakoid. So, what is a stromathylakoid? This is stromathylakoid, and these are piling of stromathylakoids. This is stroma matrix. This is appressed region, and this is non-appressed region. Exposed to chloroplast is non-appressed, not exposed to stroma matrix is non-appressed and appressed region. So, for this, all stromathylakoid membranes considered as non-appressed regions, correct. Stromathylakoids are not piled one above the other, correct. So, both A and R are correct. R is the correct explanation of A. So, first option will be the answer for this. The central idea of Hill reaction is to highlight and that what is Hill reaction? A plus H2O in the presence of sunlight, AH2 plus half wood. This A is called Hill oxidant. This is called reduced Hill oxidant. So, Hill reaction proposed that whenever there is a Hill oxidant and water is there, definitely oxygen is liberated. So, the central idea of Hill's reaction is to highlight and that existence of two photosystems is wrong. Hydrogen sulfide is hydrogen donor in sulfur bacteria is wrong. Maximum rate of photosynthesis occurs at red light is wrong. Oxygen evolved during light phase is correct. The central idea of Hill reaction is to highlight and that oxygen is evolved during light phase. So, second option. Immersion enhancement effect suggests. What is immersion enhancement effect? There are two beams of sunlight 700 and 680 playing an important role in photosynthesis. Whenever 680 is exposed to chloroplast, the rate of photosynthesis will be good. Emerson thought that 700 nanometer is still more good for photosynthesis, so he exposed a higher wavelength far red region 700 nanometers of sunlight on chloroplast. Surprisingly, he noticed a sharp decline in the rate of photosynthesis. He called it as red drop. Later, what he has done, two beams of sunlight are exposed on a chloroplast simultaneously rather than separately. Then immediately there is an increase in the rate of photosynthesis, which is called as emergence enhancement effect. So, emergence enhancement effect central idea is, emergence enhancement effect suggests that existence of two types of pigment system in thylakoid membrane is correct. PS1 and PS2. PS1 with light absorbing site at 700, PS2 with light absorbing site at 680. Photosynthesis involves two photochemical reactions. Definitely photosynthesis involves two photochemical reactions, one from PS1 and one from PS2. The two photochemical reactions are mediated by two pigment systems. Definitely two photochemical reactions are mediated by two pigment systems. They are photosystem 1 and photosystem 2, or pigment system 1 and pigment system 2. So, emergence enhancement effect suggests all the three. Existence of two types of pigment systems in thylakoid membrane. Photosynthesis involves two photochemical reactions, correct. The two photochemical reactions are mediated by two pigment systems is correct. So, answer is A, B, C, fourth option. Wrong match of the following. Ratio between chlorophyll A and chlorophyll B in LHC1 is 4:1 is correct, because 100 chlorophyll A, chlorophyll B are present in 4:1 ratio, that is 80 chlorophyll A and 20 chlorophyll B. Ratio between chlorophyll A and chlorophyll B in light harvesting complex 2 is 1:1 is correct. 250 chlorophyll A and chlorophyll B molecules are present in 1:1 ratio. Ratio between chlorophylls in PS1 and PS2 is 1:1 wrong, because in PS1 it is 100, in PS2 it is 250. It must be 10:25. Now, or 2:5. Ratio between chlorophyll A and LHC1 and LH2 is 16:25 is also wrong. Chlorophyll A in LHC1 is 80. Chlorophyll B, or what you call LHC, it is 125. So, 80, 125 will not be that. So, answer is 3. Ratio between chlorophyll A and PS2 and PS1 is always 1:1 is wrong. The third option is, during quinone cycle, PQ is reduced to PQ minus, or PQ oxidized state reduced to PQ oxidized by accepting electrons from. During quinone cycle, PQ is reduced to PQ by accepting electrons from cytochrome B. Second option. Cytochrome B provides electrons for this one. Uniformly distributed complex in both oppressed and non-oppressed regions. Uniformly distributed complex in both oppressed and non-oppressed regions is cytochrome B bar F complex. PS1 is wrong. Coupling factor complex with CF0, CF1 complex is wrong. PS2 complex is wrong. So, what is the uniformly distributed complex in both oppressed and non-oppressed regions? PS oppressed and non-oppressed means PS2 and PS1 regions. Or oppressed and non-oppressed regions is cytochrome B6 and cytochrome F complex. So, 4. C4 pathway of carbon dioxide fixation is biologically more evolved than C3 pathway. Definitely, because here there is no wastage of carbon dioxide. Total carbon dioxide is fixed in C4 pathway. So, C4 pathway of carbon dioxide fixation is biologically more evolved than C3 pathway. The fixation of CO2 requires 3 ATP and 2 NADPH2 in C3 pathway, but 5 ATP and 2 NADPH2 are required in C4 pathway. It is also correct, but it is not the correct link for that first option. C4 pathway of carbon dioxide fixation is biologically more evolved than C3 pathway is correct. The fixation of each CO2 requires 3 ATP and 2 NADPH2 in C3 pathway. 5 ATP and 2 NADPH2 in C4 pathway, both are correct but not linked with each other. So, both A and R are correct. R is not the correct explanation of A. Both A and R are correct, but R does not explains A. So, second option for this. Frenkel demonstrated cyclic electron transport in a bacterium. Frenkel demonstrated cyclic electron transport in Rhodospirillum rubrum. Rhodospirillum rubrum. So he explained this process in Rhodospirillum. So the explanation of Rhodospirillum, it is not photoautotroph, it is a photoheterotroph, it is not aerobe, it is an anaerobe. So first option is wrong. Photoautotroph and anaerobe is wrong. Photoheterotroph is correct, but it is an aerobe is wrong. Photoheterotroph and anaerobic bacteria is correct. So an anaerobic bacteria, photoheterotrophic bacteria. Photoheterotrophic means it takes energy from sunlight but carbon from organic sources. Energy from sunlight and carbon from organic sources is photoheterotroph. Photoheterotroph. So photoheterotroph and an anaerobe. Frenkel demonstrated cyclic electron transport in a bacterium, which is, what is that bacterium? Rhodospirillum rubrum, purple non-sulfur bacteria. Photoautotroph and aerobe is wrong. Photoheterotroph and anaerobe is wrong. Photoautotroph and anaerobe is wrong. Photoheterotroph and aerobe is wrong. Photoheterotroph and anaerobic bacterium is correct. So first the answer is 4. The ratio between the number of protons transported from stroma matrix to lumen through quinone cycle during cyclic and non-cyclic transport of electrons. The ratio between the number of proton ions, H plus ions, transported from stroma to lumen through quinone cycle during cyclic and non-cyclic transport of one electron. One electron always takes 2 protons. 2 electrons always take 4 protons in both cyclic and non-cyclic. So 2:2 or 4:4. The answer is 1:1. So the ratio between the number of H plus ions transported from stroma to lumen through quinone cycle during cyclic and non-cyclic transport of one electron is always 1:1, 4th option. Amount of assimilatory powers generated during light phase as per the evolution of 100 O2 molecules. When 100 O2 molecules are liberated, when 2 O2 molecules are liberated, when 2 O2 molecules are when 1 O2 molecules are liberated, 1 O2 molecule, definitely the formation of ATP will be 2 ATP and 1 NADPH2. 2 ATP and 1 NADPH2. So the answer will be 400 ATP and 200 NADPH2. Second option. 400 ATP and 200 NADPH2. Phyophytin, next assertion reasoning. Phyophytin is the primary electron acceptor as PS2 in thylakoid membrane. Definitely whenever PS2 is excited by getting sunlight from light harvesting complex 2, the excited PS2 liberate electrons which were accepted only by phyophytin. Phyophytin is the primary electron acceptor at PS2 in thylakoid membrane is correct. Phyophytin is colorless chlorophyll molecule without nitrogen is wrong. Phyophytin is a chlorophyll molecule without magnesium. The central magnesium is replaced by 2 hydrogen atoms. So the answer A is true or is false. So phyophytin is the primary electron acceptor at PS2 in thylakoid membrane. Phyophytin is colorless chlorophyll molecule without nitrogen. So A is correct or is false. That is 3 absent. Now, next assertion reasoning. Plastocyanin is the copper containing mobile electron carrier present within the thylakoid membrane. Plastocyanin is the copper containing mobile electron carrier present within the thylakoid membrane. This is wrong. Plastocyanin is the copper containing mobile electron carrier which is present in the lumen side of thylakoid membrane and moving freely here and there. That must be the answer. Plastocyanin transport electrons from cytochrome F to reaction center PS1. Plastocyanin always takes the electrons moved from water to phyophytin, water to PS2, PS2 to phenophytin, phyophytin to quinone cycle to cytochrome B6, cytochrome B6 to cytochrome F. From cytochrome F it takes electrons and these electrons are taken to PS1 is correct. So plastocyanin is the copper containing mobile electron carrier present within the thylakoid membrane is wrong. It is not present within the thylakoid membrane. It is present on the lumen side of the thylakoid membrane and moving freely here and there. Plastocyanin transports electrons from cytochrome F to reaction center of PS1 is correct. So, A is false, R is true. Fourth option. In C4 plants, to build the CO2 concentration levels in bundle sheath cells, ATP are excess utilized in. So, in C4 plants, to build the CO2 concentration levels in bundle sheath cells, excess ATP are utilized in. In C4 plants, whenever pyruvic acid moves from bundle sheaths to chlorophyll cells, bundle sheaths to mesophyll cells, in mesophyll cells, pyruvic acid converts into phosphoenol pyruvic acid by taking 2 ATP molecules excess. This is to maintain higher carbon dioxide levels in bundle sheath cells. So, in C4 plants, to build the CO2 concentration levels in bundle sheath cells, excess ATP are utilized not in PCR cycle, not in chloroplast of bundle sheath cells, not in cytoplasm of mesophyll cells, but in the chloroplast of mesophyll cells. That excess of 2 ATP in the presence of pyruvate dikinase, pyruvic acid takes and convert it into phosphoenol pyruvic acid. So, 16 question. In C4 plants, to build the CO2 concentration levels in bundle sheath cells, excess of ATP are utilized in chloroplast of mesophyll cells. So, that is fourth one. In C4 plants, during C4 pathway, oxidative decarboxylation of malic acid occurs in C4 plants, the process starts in the lumen of mesophyll cells. What happens? PEP takes carbonate ions and become oxaloacetic acid. Oxaloacetic acid sometimes undergo transamination and converts into aspartic acid. But most of the time, oxaloacetic acid moves from lumen of mesophyll chloroplast to mesophyll cells. Here it undergoes reduction and converts into malic acid. Malic acid now moves from mesophyll cells to bundle sheath cells. In the bundle sheath cells, malic acid undergoes oxidative decarboxylation to produce pyruvic acid. So, in C4 plants during C4 pathway, oxidative decarboxylation of malic acid always occurs in, not in the chloroplast of mesophyll cells, not in the mitochondria of bundle sheath cells, not in the mitochondria of mesophyll cells, but the answer is in the chloroplast of bundle sheath cells. In the chloroplast of bundle sheath cells, in C4 plants during C4 pathway, oxidative decarboxylation of malic acid occurs. That carbon dioxide liberated is taken by RUBP of the stroma matrix. C3 cycle occurs, glucose is formed in lumen of bundle sheath cells. Pyruvic acid is formed, which again moves back to mesophyll cells, utilizes 2 ATP in the presence of pyruvate dikinase, produces or regenerates phosphoenol pyruvic acid. This is a assertion reasoning. Carotenoids are absent in red algae and blue-green algae. Carotenoids are absent in red algae and blue-green algae. Carotenoids are absent in red algae and blue-green algae. Here options are changed. A is false, R is true, A is true, R is false. Phycobilins acts as accessory pigments in red algae and blue-green algae. For this, the answer must be 3. A is false and R is true. Carotenoids are absent in red algae and blue-green algae. Phycobilins acts as accessory pigments in red algae and blue-green algae is correct. Both carotenoids and phycobilins take sunlight, cannot utilize the sunlight and transport it to the reaction center. So, both carotenoids and phycobilins are popularly called as accessory pigments. Now, identify the correct ascending order of the organic compounds mentioned below based on their carbon atoms. Erythrose 4-phosphate 4 carbon, pseudoheptose 7-phosphate 7 carbon, xylulose 5-phosphate 5 carbon, dihydroxyacetone phosphate 3 carbon. So, we have to identify them in correct ascending order. The less is 3 carbon dihydroxyacetone phosphate. Next is 4 carbon erythrose 4 phosphate. Next is 5 carbon xylulose 5 phosphate. Last is 7 carbon pseudoheptose 7 phosphate. So, these are all formed during regeneration of RUBP in C3 cycle. So, identify the correct ascending order of the organic compounds mentioned below based on their number of carbon atoms. Ascending order because less is 3, dihydroxyacetone phosphate. Next is 4, erythrose 4 phosphate. Next is 5, xylose 5 phosphate. Next is 7, pseudoheptose 1 or pseudoheptose 7 phosphate. So 4, 1, 3, 2 will be the answer. That is the first option. Now, this is a 3-column matching form. We have to identify the correct ones. Oxaloacetic acid converts into malic acid in the presence of malic dehydrogenase. Oxaloacetic acid converts into malic acid in the presence of malic dehydrogenase is correct. Pyruvic acid converts into phosphoenol pyruvic acid in the presence of pyruvate kinase is wrong. It is pyruvate dikinase. So wrong. Pseudoheptose 1,7 biphosphate converts into pseudoheptose 7 phosphate in the presence of phosphatase is correct because phosphatase removes the phosphate from pseudoheptose. It is a hydrolytic enzyme. Xylose 5 phosphate converts into ribulose 5 phosphate in the presence of lyase is wrong. In the presence of epimerase, xylose 5 phosphate converts into ribulose 5 phosphate. So the two correct are 1 and 3. Oxaloacetic acid becomes malic acid by malic dehydrogenase is correct. Pyruvic acid in the presence of phosphoenol pyruvic acid, not pyruvate kinase, it is pyruvate dikinase. Pseudoheptose 1,7 biphosphate becomes pseudoheptose 7 phosphate in the presence of phosphatase is correct. Xylose 5 phosphate converts into ribulose 5 phosphate not by lyase but by epimerase. So answer for this is third option 1 and 3. Mismatch in the following. Ratio between chlorophylla of antenna of chlorophyll HC1 and LH2 is 16:25 correct. Ratio between chlorophyll B of antenna of LHC1 and LH2 is 4:25 is correct. Ratio between chlorophyll A and chlorophyll in the LHC and LHC1 is 9:14 is wrong. Ratio between the chlorophylls of the antenna of LHC1 and LH2 is 2:5 is correct. So here the wrong is ratio between chlorophyll A and chlorophyll B in the LHC1 and LHC2 is 9:14 is mismatch, wrong. How many ATP molecules are used in excess during the synthesis of one hexose molecule in C4 plants when compared with C3 plants? In C3 plants, for the fixation of 1 CO2, 3 ATP and 2 NADPH2 are utilized. For fixation of 6 CO2, 18 ATP and 12 NADPH2 are used. This is in C3 plants. In C4 plants, for 1 CO2, ATP utilized is 5 ATP and NADPH2 number is same, 2 NADPH2. For 6 CO2, 30 ATP and 12 NADPH2. So in C3 plants and C4 plants, the utilization of NADPH2 is same, 12 NADPH2. But coming to the ATP, here 18, here 30. So the difference is 12 ATP. So 12 ATP are, how many ATP molecules are used in excess? Are used in excess in C4 plants when compared with C3 plants. How many ATP molecules are used in excess during the synthesis of one hexose molecule in C4 plants compared to C3 plants is 12 ATP molecules. This is a matching. Peter Mitchell, Robert Hill, Robert Emerson, Rubin and Kamen. Peter Mitchell is 4. Proton gradient is the driving force for ATP synthesis. Proton gradient is the driving force for ATP synthesis is chemiosmotic theory proposed by Peter Mitchell. Robert Hill, isolated chloroplasts release oxygen in the presence of light and a oxidant, but absence of carbon dioxide. So Robert Hill, isolated chloroplasts release oxygen in the presence of light and an oxidant, first option. Robert Emerson, existence of two photochemical reactions, that is PS1 and PS2, or pigment system 1 and pigment system 2 are discovered by Robert Emerson. Rubin and Kamen used isotope to confirm release of oxygen from water. They used O18 isotope in H2O. Whenever oxygen is liberated, it contained 18. So proving that the oxygen that is coming during photosynthesis is not coming from carbon dioxide but coming from water. So Peter Mitchell, proton gradient is the driving force for ATP synthesis. Robert Hill, Isolated chloroplasts release oxygen in the presence of light and an oxidant, that is called photolysis of water or Hill reaction. Robert Emerson, existence of two photochemical reactions PS1 and PS2, presence of two pigment systems by Robert Emerson. Rubin and Kamen used isotope to confirm the release of oxygen from water. So A4, B1, C3, D2, third option. Unit of assimilatory power. Unit of assimilatory power means one assimilatory power. One assimilatory power which can fix one carbon dioxide is called unit of assimilatory power. 1 ATP and 1 NADPH2 is wrong. 18 ATP and 12 NADPH2 not for 1, for 6 carbon. 12 ATP, 12 NADPH2 is wrong. So answer for this is 3 ATP and 2 NADPH2. So unit of assimilatory power is 3 ATP and 2 NADPH2. Arrange the following in ascending order based on the number of hydrogen atoms per molecule. Based on the number of hydrogen molecule per molecule in ascending order. Ethyl alcohol C2H5OH, so only 5. Pheophytin C55H74, so 74. Glucose C6H12O6. Chlorophyll A C55H72. Carotene C40H56. According to this, first always 1, then 3, glucose, ethyl alcohol 5, glucose 6, then 5, carotene, then 4 is chlorophyll A, then 2, pheophytin, because H74. So arrange the following in ascending order based on the number of hydrogen atom per molecule: ethyl alcohol, pheophytin, glucose, chlorophyll A, carotene in photosynthesis. Answer is 1, 3, 5, 4, 2. 1 ethyl alcohol 5, 3 glucose 6, 5 carotene C40 H56, then chlorophyll A, and finally pheophytin. So 1, 3, 4, 5, 4, 2 is the answer. First option. In C4 plants, primary carboxylation is catalyzed by. Primary carboxylation means takes place in the lumen of mesophyll cells. What is that primary carboxylation? Phosphoenol pyruvic acid taking carbon dioxide and forming oxaloacetic acid, and carbon dioxide in the form of bicarbonates. So RUBP carboxylase is wrong, pyruvic decarboxylase is wrong, carboxylase is wrong. So pepcase means PEP carboxylase, second option. C3 plants differ from C4 plants in all aspects except very high carbon dioxide compensation point is found in C3 plants. Absence of cranch anatomy and chloroplast dimorphism is absent in C3 plants and found in C4 plants. High photorespiratory activity is found in C3 plants, not in C4 plants. So C3 plants differ from C4 plants in all aspects except means the common concept of C3 and C4 plants is utilization of ATP and NADPH2 for CO2 reduction is same in C3 plants and C4 plants, because very high carbon dioxide compensation point is shown by C3 plants, not by C4 plants. Absence of cranch anatomy and chloroplast dimorphism is found in C3 plants but present in C4 plants. High photorespiratory activity is found in C3 plants, absent in C4 plants. So common feature is ability of utilization of ATP and NADPH2 for CO2 reduction. So second option. This is an assertion reasoning. Two types of photosynthetic carbon reduction pathways are identified in C3 cycle and C4 cycle. Two types of photosynthetic carbon reduction pathways are identified as C3 cycle and C4 cycle. C3 cycle is one photosynthetic carbon reduction. C4 cycle is another photosynthetic carbon reduction. Two types of carbon reduction pathways of photosynthesis are named after the first stable compound formed of carbon reduction. When 3-phosphoglyceric acid is formed, it is called C3 cycle. Whenever oxaloacetic acid is formed, it is called C4 cycle. So two types of photosynthetic carbon reduction pathways are identified as C3 cycle and C4 cycle is correct. Why it is identified as C3 and C4 cycle means the two types of carbon reduction pathways of photosynthesis are named after the first stable compound of carbon reduction is correct. So both A and R are correct. R is the correct explanation of A. This is another assertion reasoning. The two carboxylation of C4 plants are spatially separated. One occurs in the lumen of mesophyll, the other occurs in the bundle sheath cells. PEP carboxylation occurs in the mesophyll cells and RUBP carboxylation occurs in the bundle sheath cells in C4 plants. So A and R are correct. R is the correct explanation of A, first option. This is a matching. Match the following with reference to Calvin cycle. RUBP, glyceraldehyde 3-phosphate, phosphoglyceric acid, starch. RUBP, primary CO2 acceptor, second option. Primary CO2 acceptor. Glyceraldehyde 3-phosphate, almost immediate end product. Once the glyceraldehyde 3-phosphate is formed, carboxylation and reduction both are over. So, immediate end product, fourth option. PGA. PGA is first stable product. First stable products in C3 cycle is always phosphoglyceric acid. So, first stable product. Starch. Ultimate end products. Ultimate end products of Calvin cycle is starch. So, RUBP, primary CO2 acceptor. G3P, immediate end product. PGA, first stable product, and starch, ultimate end product. A2, B4, C3, D1, first option. What is not required for carboxylation in C3 cycle? What are not required for carboxylation in C3 cycle? Rubisco is required, PGA is not required, H2O is required, H2O is required, ATP is not required, RUBP is required, O2 is not required, CO2 is required. So, all these are required. Which are not required means B, D, F. What is B? Phosphoglyceric acid. What is D? ATP, and F, oxygen. These three are not required for carboxylation in C3 plants. C3 plants does not require phosphoglyceric acid, ATP, and oxygen. B, D, F. This is an assertion reasoning. Starch is also synthesized in the chloroplast when sucrose synthesis does not keep pace in the Calvin cycle. When sucrose synthesis does not keep pace in the C3 cycle, starch is also synthesized in the chloroplast is correct. Accumulation of trioses in cytosol prevents the further export of triose from chloroplast into cytosol. Accumulation of trioses in cytosol prevents the further export of trioses from chloroplast to cytosol is wrong. So, A is correct, R is wrong. Third option. Common substrate product in transketolation reaction of Calvin cycle. Substrate and product in transketolation reaction of Calvin cycle respectively. Substrate, whenever fructose 6-phosphate combines with glyceraldehyde 3-phosphate in the presence of transketolase enzyme, by transketolation 5 carbon compound xylose 5-phosphate is formed, 4 carbon compound erythrose 4-phosphate is formed. Again, when pseudoheptose 7-phosphate combines with glyceraldehyde 3-phosphate, definitely again by transketolase enzyme, transketolization, xylose 5-phosphate and ribose 5-phosphate are formed. So, common substrate and product in transketolation reaction of Calvin cycle respectively are fructose 6-phosphate and xylose 5-phosphate is wrong. Pseudoheptose 7-phosphate and ribose 5-phosphate is wrong. Glyceraldehyde 3-phosphate will be the substrate, xylose 5-phosphate will be the product. So, definitely the answer is third one. Glyceraldehyde 3-phosphate, xylose 5, DHAP and xylose 5-phosphate is wrong. So, third option is correct. Now, match the following with reference to Calvin cycle. List 1, carboxylation, phosphorylation, isomerization, reductive dephosphorylation, last step of regeneration phase, first step of Calvin cycle, last step of reductive phase, first step of reductive phase, first step of regeneration phase. A2, carboxylation, first step in Calvin cycle. First step in Calvin cycle is carboxylation. B4, phosphorylation, first step in reductive phase. Phosphorylation is the first step in reductive phase. C5, isomerization, first step in regeneration phase. First step in regeneration phase is conversion of glyceraldehyde 3-phosphate into dihydroxyacetone phosphate. 3 remains as it is. 2 glyceraldehyde 3-phosphate get isomerized to form regeneration DHAP. Next 3, reductive dephosphorylation, last step of reductive phase. Reductive dephosphorylation is the last step of reductive phase. So, A2 before C5D3. First option. Which of the following is dissimilar type of sugar from others? Which of the following is the dissimilar type of sugar from the others? Which of the following is dissimilar type of sugar means xylulose is 5 carbon compound, ribose is 5 carbon compound, ribulose is 5 carbon compound, erythrose is 4 carbon compound. So xylulose, ribose, ribulose belongs to the same group. Erythrose is 4 carbon compound. So definitely which of the following is a dissimilar type of sugar from the others means erythrose, 4 carbon compound. Number of G3P and dihydroxyacetone phosphate required to form 3 molecules of RUBP respectively in C3 cycle. Number of glyceraldehyde 3-phosphate and di-HAP required to form 3 molecules of RUBP respectively through C3 cycle is, out of 5 molecules of glyceraldehyde 3-phosphate in stroma matrix, 3 molecules of glyceraldehyde 3-phosphate remains as it is. 2 molecules of glyceraldehyde 3-phosphate converts into 2 molecules of dihydroxyacetone phosphate. So, number of glyceraldehyde 3-phosphate molecules and dihydroxyacetone phosphate molecules for regeneration of all the 3 molecules of RUBP will be 3 and 2. So, number of glyceraldehyde 3-phosphate molecules and dihydroxyacetone phosphate molecules to form 3 molecules of RUBP respectively through C3 cycle is always 3 and 2. Dual nature of ribosco results in decarboxylation of Calvin cycle in C3 plants is wrong. Oxygenation reaction in mesophyll cells of C4 plants is wrong. Carboxylation of RUBP in agranule chloroplasts of C4 plants is correct. Oxygenation of RUBP in C3 plants is correct. So, dual nature of ribosco can be results in carboxylation of RUBP in acarboxylation of chloroplast, agranule chloroplast of C4 plants is correct. Oxygenation of RUBP in C3 plants is correct. Decarboxylation in Calvin cycle of C3 plants is wrong. So, answer is 3 and 4. Recycling of the 3 carbons of serine to Calvin cycle involves. Recycling of 3 carbons of serine to Calvin cycle involves reduction of hydroxypyruvate wrong. Reduction of hydroxypyruvate is correct. Oxidative decarboxylation of glycine is wrong. Phosphorylation of glycerate is correct. Dephosphorylation of phosphoglycolate is wrong. Deamination of serine is correct. Oxygenation of RUBP is wrong. Recycling of 3 carbons of serine to Calvin cycle means it is related to photorespiration. E, A and C. First option. During photorespiratory path, reduction of NAD and oxidation of NADH occur respectively during the formation of. During photorespiratory path which occurs in chloroplast, peroxisomes, mitochondria, reduction of NAD plus and oxidation of NAD plus occurs respectively during the formation of serine and glyceride. When serine is formed from 2 molecules of glycine, reduction of NAD takes place. Whenever hydroxypyruvate converts into glyceric acid, their oxidation of NADH2 occur. So, during the formation of serine and glycerate, during photorespiration, reduction of NAD and oxidation of NAD occurs respectively. This is an assumption reasoning question. C4 pathway is biologically more evolved than C3 pathway. Definitely C4 pathway is more evolved than C3 pathway. Reason: C4 plants can perform active photosynthesis even under water stress conditions. So, C4 plants grow in water stress environments. Almost all tropical plants show C4 pathway, and these plants can perform active photosynthesis even under water stress condition is correct. So, why C4 pathway is biologically more evolved than C3 pathway? Because C4 plants can perform active photosynthesis even under water stress conditions. So, A and R are correct. R is the correct explanation of A, first option. Wrong match of the following. PEP, primary acceptor of CO2 in beta carboxylation pathway is correct. PEP carboxylation enzyme found in all chlorophyll cells of C4 plants is wrong. Oxaloacetic acid, a 4-carbon dicarboxylic acid formed as first stable compound in C4 pathway is correct. Malic acid, reductive product of oxaloacetic acid is correct. So, wrong match is PEP carboxylase carboxylation enzyme found in all chlorophyllous cells is wrong because it is absent in bundle sheath chloroplast. So, it is present only in the lumen of mesophyll chloroplast. That is why wrong match is PEP carboxylase carboxylation enzyme found in all chlorophyllous cells of C4 plants is wrong match. This is another assertion reasoning. In an atmosphere having higher concentration of oxygen, C3 plants gain less dry weight from C4 than C4 plants during daytime. This is regarding photorespiration. C4 plants 0 photorespiration occurs, C3 plants maximum photorespiration occurs. When oxygen is more, C3 plant takes more oxygen, and definitely there is less in dry weight in C3 plants. So, in atmosphere having higher concentration of CO2, C3 plants gain less dry weight than C4 plants during daytime. In higher concentration of CO2, photorespiration occurs in C3 plants, whereas photorespiration is not detectable in C4 plants. So, A and R are correct. R is the correct explanation of A. This is regarding photorespiration in C3 and C4 plants. The number of carboxylation and decarboxylation occurs respectively in C4 pathway of carbohydrate synthesis. In C4 pathway, carboxylations are 2, where one is the lumen of mesophyll cells, other in the chloroplast of bundle sheath cells. So, lumen of chlorophyll cells, how? Phosphoenol pyruvic acid get carboxylated to form oxaloacetic acid. How in bundle sheath cells? RUBP takes the carbon dioxide liberated from malic acid and performs C3 cycle. Carboxylations are 2, decarboxylations only 1. Where this occurs? Oxidative decarboxylation of malic acid occurs in the bundle sheath cells of C4 plants. So, answer is 2 and 1, third option.